Yeah, good morning. And first of all, a warm thank you to the scientific committee for the invitation to speak here today. It really is a great honor. And following Glenda's excellent talk on vaccines, I will try to provide an overview of immunotherapies towards cure and remission. And these are my disclosures. The fundamental problem is, of course, that antiretroviral therapy is highly effective in suppressing HIV, but although they're so effective, we also know that once treatment is stopped, there's a rapid virus rebound. And this is happening because HIV establishes a latent infection in long-lived cells. This is the latent reservoir, which remains the primary barrier to a cure and therefore the target for curative strategies. There are two overarching goals in cure research. One is focused on the complete elimination of all infected cells, leading to what we usually refer to as eradication. And the other approach is focused on reducing the pool of latently infected cells and then relying on immune effector mechanisms to maintain control of whatever is left. And this reduce and control strategy could be an approach to achieving remission. Immunotherapies could play a key role in achieving remission, primarily by inducing durable and potent anti-HIV immune surveillance. Listed here are some of the most important immunotherapies that are currently under active investigation. And these include interventions that primarily work by enhancing cytotoxic T cells, by inducing innate immune responses such as NK cells, therapies that primarily work through antibody-mediated mechanisms, as well as interventions that are capable of targeting several arms of the immune system. As Glenda touched on, there are potential synergies between immunotherapies for cure and research into vaccines. First of all, the immune responses induced by any efficacious vaccine will of course be highly relevant to that potent and durable immune surveillance, which we know is required to achieve remission. And conversely, any effective immunotherapy may also provide relevant clues to targets for disease-modifying or even preventive vaccines. And for both lines of research, I think it's highly important that we achieve a very thorough understanding of the mechanisms that underlie elite control and also post-treatment control. There are several questions that are quite important and which are surrounding HIV immunotherapies. First of all, there's a fundamental issue of whether a given intervention can provide a durable effect or if there's a need for ongoing treatment. And ideally, to achieve cure or remission, we do need time-limited interventions that are capable of inducing long-lasting control. Another important question is whether some therapies may have an enhanced effect if they're delivered in the setting of active HIV expression. This may be the case for therapies such as immune checkpoint antibodies, which primarily modulate T cell function in the setting of antigen recognition. And I think this difference in effect between suppressed and non-suppressed infection is quite important to consider for each of the therapies we're looking at here. It's also important to consider which of these therapies, based on their interaction with the immune system, might make ideal candidates for combination strategies. And finally, I do find it useful to distinguish between interventions that constitute viable clinical strategies as opposed to drugs that are probably too toxic, but where it's still useful to achieve an understanding of how interrupting or modulating a given biological pathway might impact HIV persistence. So broadly neutralizing antibodies, as mentioned just before, this is probably the most heavily investigated type of intervention in cure research recently. And in the first, these are antibodies that have been isolated from a minority of infected individuals. And due to recent advances, these antibodies can now be produced at a scale that have allowed for their clinical application. There is a growing number of BNAbs that have now been identified, and these are usually categorized according to their binding site on the HIV envelope trimer. And this includes the CD4 binding site antibody 3BNC117, as well as the V3 glycan site antibody 10-1074. I'll get back to those two antibodies in a minute, but generally speaking, many of these BNAbs are now passing through clinical trials to assess their potential for using them as treatment, prevention, or cure. In the first study testing a combination of two BNAbs during ART interruption, Mendoza and colleagues showed that 3 infusions of 3-BNC 117 and 10-1074 could maintain viral suppression for up to 30 weeks in people interrupting ART, and a more recent study also indicated a potential minor impact from these antibodies on the intact HIV reservoir. Interestingly, in the study by Mendoza and colleagues, 2 of 9 individuals receiving dual BNAbs also displayed prolonged biological control even way after the BNAbs had decayed to below the level of inhibition. This and other findings have raised the question whether BNAbs, in addition to neutralizing free virus, may also have an effect of boosting T-cell function, which has been referred to as a vaccinal effect. This question is still somewhat controversial, but using samples from this study, Daniel Kaufman and his team recently addressed this question and found that in the weeks following BNAb therapy, there was a significant increase in the frequency of HIV-specific T-cells producing antiviral cytokines in response to peptide stimulation, thus indicating a potential vaccinal effect. Another question is whether we're targeting the reservoir at the right time. When you look back, in fact, most cure studies have tested interventions that are given in the setting of long-term suppression by ART, but it has also been shown that the composition of the latent reservoir is primarily determined or frozen at the time when ART is initiated. So it is conceivable that immunotherapies, especially those relying on antigen recognition, may have a greater efficacy if delivered in this viremic phase at ART initiation. This randomized controlled trial of 3-BNC 117 and romidepsin given at ART initiation investigated precisely this question. This is a study that has been led by Ole Søgaard and was presented at CROI earlier this year. And the study furthermore provided a unique opportunity to address the potential vaccinal effect from BNAbs. So in this study, they enrolled 60 people that were all newly diagnosed, and participants were randomized to either ART alone or to ART in combination with either 3-BNC 117, romidepsin as a latency reversing agent, or the combination of the two. Participants were then maintained on suppressive ART for 1 year, after which 2 key outcome measures were quantified: the impact on the intact HIV reservoir and the time to viral rebound during an optional treatment interruption. And it is worth noting that in the groups receiving 3-BNC, only 53% and 63% of participants harbored viruses that were sensitive to 3-BNC before the intervention. When ART was interrupted, there was a mixture of outcomes, with some individuals rebounding early, but also a fair number displaying enhanced control. But the really fascinating finding was that enhanced biological control was primarily, almost exclusively happening among those harboring 3-BNC 117 sensitive viruses in their reservoir. And this included 1 individual who has now maintained undetectable viral load for more than 4 years after stopping ART in the study. To address whether this might have been achieved through an effect of the BNAbs on T-cell function, they quantified the frequency of HIV-specific CD8 T-cells using the activation-induced marker assay, and this demonstrated that as compared to the ART alone group, 3-BNC 117 recipients maintained a significantly higher frequency of HIV-specific T-cells, and this was primarily driven again by those harboring 3-BNC 117 sensitive viruses. So in conclusion, this demonstrates, or at least strongly indicates, that 3-BNC at ART initiation could lead to a sustained boost in HIV-specific T-cell function and was associated with enhanced control after stopping ART. Therapeutic stimulation of Toll-like receptors is another strategy that's being investigated as an approach to targeting HIV persistence, and most studies have focused on either the TLR9 agonist, lefitolimod, or the TLR7 agonist, visatolimod. Both of these bind to endosomal receptors in plasmacytoid dendritic cells and B cells and induce a broad response that includes secretion of type 1 interferons, induction of interferon-stimulated genes, and activation of cytotoxic NK cells. And there's been a range of studies testing a combination of a TLR7 agonist, a BNAb, and/or therapeutic vaccination in rhesus macaques infected with SIV or SHIV. And these have produced quite remarkable results, including the 2 front studies here that demonstrated prolonged virological control in more than half of the monkeys receiving combination therapy. Achieving remission in human infection, though, is likely to be a greater challenge, as there are, of course, important differences between rhesus macaques and humans, including host-specific differences in their genetic and immunological constitution. Depending on the utilized virus strain, there may also be differences in virus pathogenicity between HIV and HIV. And importantly, initiating ART extremely early after infection, as was done in many of these macaque studies, is of course usually not possible during human infection. A couple of clinical trials, including the two shown here, have also tested the administration of the TLR7 or 9 agonist in people with HIV. And these studies have demonstrated their ability to activate cytotoxic NK cells, dendritic cells and T cells, and induce plasma interferon alpha and induction of interferon-stimulated genes. In the study on the right, conducted among HIV controllers on ART, there was also a modest delay in time to rebound after treatment with the TLR7 agonist visotolimod. And several ongoing clinical trials are now testing the TLR7 or 9 agonist in combination with either a vaccine, dual BNAbs, or both. And the outcome of these studies, I think, will be highly important to assessing whether the encouraging data seen in rhesus macaques might be replicated to some extent during human infection as well, and whether therapeutic stimulation of toll-like receptors therefore might play a role in immunotherapeutic strategies towards remission. Immune checkpoint proteins, over the past decade or so, we have seen that immune checkpoint proteins play a role for HIV persistence. We now know that latent HIV is enriched in cells expressing some immune checkpoints, and that blocking these can activate HIV from latency. It's also clear that increased expression of immune checkpoints mediate T-cell exhaustion during chronic HIV infection. There is some evidence that blocking PD-1 can enhance at least SIV-specific T-cell function and lead to enhanced control in macaques. But there is still very limited clinical data regarding the impact of immune checkpoint blockade on HIV-specific T-cell function and the latent reservoir in people with HIV. And the few data that we have primarily stems from studies in people who are receiving these antibodies due to advanced malignancies. Some immunotherapeutic agents are capable of targeting both innate and adaptive immune effector cells. IL-15, for example, is a powerful immune activation cytokine that promotes the expansion and activation of both CD8 and NK cells. In HIV-infected rhesus macaques, the IL-15 superagonist, N803, induced proliferation and activation of virus-specific CD8 T cells, and also promoted their migration into B-cell follicles, which we know is a site of HIV persistence. The first clinical trial of N803 in people with HIV was published earlier this year in Nature Medicine, and this study demonstrated safety as well as activation of T and NK cells. In this study, they did not find any evidence for an expansion of HIV-specific CD8 T cells, but they noted a gradual decrease over the course of the study up until 6 months in the frequency of cells harboring inducible HIV, which is of course an interesting finding, but larger studies are required to fully understand the impact of this molecule on HIV persistence, on immune control of the latent reservoir, and whether there may be synergistic effects when combining this with other immunotherapies such as BNAbs. Another compound that appears to be able to activate both innate and adaptive immunity is pomalidomide. This is an immunomodulatory drug that's approved for the treatment of multiple myeloma and age-related KS. And over the past few years, we've carefully examined the immunological effects of pomalidomide in the lab, and we saw some interesting effects. We found that ex vivo treatment with pomalidomide could expand the population of cytotoxic NK cells and also enhanced NK cell killing of autologous in vitro HIV-infected CD4 target cells. We also find that adding pomalidomide to cell culture in the presence of an immunodominant peptide significantly expanded the frequency of the tetramer-positive HIV-specific CD8 T cells, as well as their killing of HIV peptide-loaded target cells. All this work has been done by an excellent PhD student, Rachel Pascoe, who's presenting her work at this conference, and for more information, please go see her poster, which is listed here. So this is bringing me to my conclusions. that some combinations of immunotherapies look very promising in animal models, and encouraging data are now emerging in human trials as well. It was recently shown that BNAb therapy at the time of ART initiation could provide a sustained boost to HIV-specific T-cell function and even improved ART-free control. So this is raising the question whether immunotherapeutic targeting of the reservoir at the time of ART initiation, or maybe during viremia, might impact the course of HIV infection and increase the likelihood of achieving remission. This would be a fantastic result, but even if successful, immunotherapies in their current form are unlikely to lead to complete eradication, and this is really leaving important societal and economic issues to be considered even if remission is achieved. And finally, I think it's worth emphasizing that all these therapies are part of a bigger picture and just one of several approaches currently being tested to achieve HIV cure. And finally, I just wanted to acknowledge a large group of really terrific collaborators from around the globe, many of whom have contributed to the results reviewed here. A word of thanks to our funders, and especially a huge thank you to the people with HIV who have contributed their time and effort by participating in research studies to advance our knowledge of the virus. Thank you.